Okay, I'm not going to move back. I think you have seen me. Otherwise, we have plenty of time afterwards to look after at each other. Thanks a lot for the invitation. If I sometimes look the other side, that has nothing to do with you, is because like I have two screens, okay? And I always forget that I should talk to a green light instead of looking at my screen. So it's a bit odd. So thanks a lot for inviting me. I think this was planned for a very long time ago, and then like the world has come to complete chaos, and I think we have still complete chaos from a day to day. So what better to do than use an evening and talk about bees, because that's hopefully the passion that we share, or all the ones that join us tonight. And if you're puzzled by like my accent, and you wonder from which part of the US that comes from, I have to disappoint you. This is where I grew up. That's in Switzerland. That's the house where I was born. I was actually born at home. We didn't have a hospital close by. So in that room up here, I was born in the countryside. And the house was my grandmother's, and she lived there. And the house was built 1901. And my grandfather was a painter, so he painted this house. And the house is still in the family, so my sister lives in there. And for me, I grew up with my grandmother in that house, and you can see that she had a passion for her garden. So she planted a lot of fruit trees, and that had a specific relevance for me because like I hate winter. I can't stand winter. One reason to end up in California. Winters in Switzerland are cold and very dark, and for me, too long. And what happened, like, and what really cheered me up is spring, right? So February, March, what happened is the trees of my grandmother became to bloom, or went into bloom, and all of a sudden they were filled with insects, especially with bees. You can see here a bumblebee, but we had like the entire variety of bees that were available there. I found several bumblebee nests, for example, in that garden every year. And that's really interesting because I don't think I've seen a wild honeybee nest for the last 15 or 20 years. So I refer to that as the golden age of bees. They were around, I could hear them, I could smell them. They signaled to me the end of the winter that I really hated. And so it was probably not like surprising that I kept that interest in animals and in bees. But by the time I had finished my studies at the university in Zurich, there were these first reports going around about like the decline of bees, the death of bees. It started with beekeepers reporting real problems to keep bees alive. But eventually it also kind of became apparent that like other bee species, native bees, bumblebees and so on, like that, they also suffer from that. So when I was looking for a PhD, I really wanted to save the bees. And I got a PhD position at the Swiss Federal Institute for Technology. And I was very motivated. My supervisor basically gave me a really interesting study topic. I was not necessarily the one that I expected to save the bees, but here we go, right? So basically what he said is like that in order to save the bees, we really have to understand of like what's bugging them, and also like how the bees could potentially kind of react to them and could be saved from these threats. And one of the major threats that bees clearly have are parasites and pathogens. Honeybees, classic example, they have incredible large amounts of different diseases, but it's the same for bumblebees. And at that time, there was an interesting idea around, but there was very little data to kind of confirm that. And that idea was that like the genetic diversity that you find within a hive, that is a really good protection against diseases. And the way this is accomplished by bees would be that like you have a queen, a bumblebee queen here, as you can see, and she mates with a male. And of course, if she mates with more than a single male, if she mates with multiple males, and she sires offspring from these multiple males, that increases the genetic diversity among bees, right? and you probably all know that like honeybees are really going for it when it comes down to multiple mating. The queens, the honeybee queens have mating frequencies between like 16 and 90 males, right? So they seem to be really kind of interested in that genetic diversity. Anyway, so he wanted me, my supervisor wanted me to kind of generate data to kind of show whether that's the case. And I started to do some experiments. And of course, like the most easy way to kind of approach this is if you kind of create hives that have different levels of genetic diversity. So you have a queen that mates only with one male, and then you have other queens and they mate with multiple males. And I really tried pretty much everything. The queens mated with the first male, but then I had really problems to kind of find any queen that would ever mate again. So we decided to take like that into our own hands, and I developed an artificial insemination technique for bumblebees. It's very similar to the one that is used in honeybees. It's a bit more complicated, but we eventually got that to work. So you have like here, you have the sexual tract of a bumblebee, and you enter a small kind of glass capillary that contains the semen, and eventually here in that red dot here, that signifies the spermatheca, the sperm storage organ, so that's where the sperm is stored. So with that, like if you have full control over that, you can then kind of manipulate how much genetic diversity you would have in a beehive. And we basically tested that under field conditions. And what we found is that indeed, like multiple mating increases the genetic diversity, and that has quite a substantial effect on the performance of the colony. And so that was the kind of first indication that like genetic diversity is a real important driver. And I think in between now, over the last couple of years, the same effect has also been shown in honeybees. So it seems to be the case in a number of different bee species. So that was my entry into the world of bees and parasites. The interesting thing is when I started my PhD, that the tradition in the Swiss system is that like normally the first year PhDs get a third year PhD student allocated to, and that third year student then basically shows the rookie like, you know, how to catch bees, how to breed bees, how to keep them, right? And in my case, that was a third year female PhD student. And this kind of relationship kind of was quite successful because it's still in place today. So I met my wife. At that point, she was actually my supervisor. Since these times, we basically travel the world together, and we both have like this incredible passion about bees. And eventually, like, we expanded our little family. We have 2 boys now. They're a little bit older than on the picture here. They are now 12 and 16, and both have a passion for bees as well. My younger son wants to become like an insect researcher, and the older one is actually really interested in honeybees as well. So with that, we kind of moved out of Switzerland at some point. We got invited to join a university in Western Australia in 2005. So that was a long way from home, from Switzerland, but there was a really kind of good reason kind of to move to Australia, because it has very unique landscapes, as you can see here. It's a very lonely place. The next bigger cities in our case, this was Perth in Western Australia, is like 4 or 5 hours flight time away. That's either Singapore or Sydney. So it's very isolated, but that had tremendous effects on the bees there, because it's so isolated that no one brought bees there, and they have a complete ban of bringing in any bees. So these are really healthy bees. They don't have the hive beetle, they have no varroa mites, they have no deformed wing virus, they have very few parasites. Because it's so isolated, there's very little agriculture happening in these areas. So bees are not really exposed to enormous amounts of pesticides. The climate is really nice to bees. So we can study in Western Australia really healthy bees. So we can basically define like, this is the type of hive that everyone wants to have. And we got invited to Western Australia to initiate like a hub of research, and we called that the Center for Integrative Bee Research. And as soon as you kind of study bees, I think it doesn't take a lot of time, especially when you're interested in honeybees, that like you realize that if you really want to make like any reasonable impact, you actually have to involve the beekeepers and the guardians of these animals as well. So we started to build a very close relationship with the local beekeepers, both with the commercial ones. Here, for example, you can see Barbara marking queens together with commercial beekeepers. But we also kind of build a very close relationship with hobbyist beekeepers. And when we arrived in Perth, the Hobby Beekeeper Club was a bit kind of at the brink of kind of shutting down. I think they had like 50 members. And there was a big push in Western Australia, and especially in Perth. Beekeeping became quite in fashion. There was increasing interest in bees. And by the time we left Western Australia, that the club actually had 1,000 members. So it kind of really increased in size. What we hooked up with was also a commercial kind of enterprise, which is called Better Bees. It's one of the biggest breeding programs in the world, where like each of these beekeepers maintains 3 to 4 lines. So we have something around 30 lines that were set up more than 30 years ago. And they still maintain these bees. And the beekeepers were quite interested because, as you have seen before, I started to study effects of genetic diversity on bee health. But they were also interested because I was able to artificially inseminate bees and help them with their breeding efforts. Now, the way the Better Bees program is run is very impressive. Each of these beekeepers assesses these lineages over a year and selects the best running beehives. And that is based on about 20 to 25 different kind of factors that they basically continuously kind of monitor. And they then produce males, and they go after queens from these hives. They put these queens on a big truck. So you can see the truck here. And then the truck is put on a barge, and the barge is traveling about 15 miles offshore to an island. And on that island, the bees are then allowed to open mate. So the virgins are released, and they have the males from the breeding program. So you have a closed breeding program that is happening on that island of Better Bees. And that is, as I said, this is very impressive to see. It takes quite some organization from the beekeepers to get this up and running, because every beekeeper needs to have the queens and the males ready at this specific point in time. And they have to all transport these bees to that island, and some of them are coming from hundreds of miles away, right? They're not all around Perth. Now, these bees are really remarkable. We were allowed to use them for our research, and we basically did that quite a lot, because the performance of these bees is absolutely remarkable. First of all, because what I said before, the bees in Western Australia are already quite healthy. There's actually a complete ban within Western Australia of any chemical treatment. And that might be easier to kind of do when there is no varroa. But Western Australia has other diseases. For example, American foulbrood is quite widespread. So you cannot treat American foulbrood with any chemicals. And that means the stock that they maintain in their breeding program has levels of tolerance against American foulbrood as well. And during the time we were actually working in Australia, over these 15 years, we maintained an apiary of 60 plus hives. And in 15 years, we lost 2 hives. And that's probably what we lose here in 1 month, right? So how are these bees otherwise performing? I don't know what you get out of your hives here in California on a yearly basis. The hives of the Better Bee program produce up to 650 pounds of honey per year. So they're really going for it. They're extremely nice to manage. Our queens typically lasted 2 to 3 years. So it's like, for me, like an indication is that what bees, if they're healthy, what they're actually kind of capable to do. And that's quite impressive to see. What we also did with the beekeepers is we kind of set up quite some outreach program, because in the beginning it was clear it would be really helpful if we promote beekeeping, especially responsible beekeeping. And we also sense there's quite a lot of interest in the broader public about bees and making honey, for example. So this is one example here. We set up Honey Week, which runs, I think, pretty much at the moment again in Australia. So it's a week where a lot of bee activities take place around Perth. And it, basically, there's at the end of that week, there's the Honey Festival. So these are people that have organized the Honey Festival, which is then kind of visited by a couple of thousand people each year. We were also involved in the making of a documentary, which was kind of triggered by the release of the Bee movie. I don't know whether you have seen that. I think it was the only time I was close to being thrown out of a movie theater, because my enthusiasm for that Bee movie was really limited, because it's just so inaccurate. And when we do outreach, we get continuously kind of bombarded with facts from the Bee movie, which we then have to kind of correct. And I got a bit annoyed by that. So we kind of did a movie where we thought we need to get the scientific facts and the life of the bee correct, also the way the beekeepers are depicted in there. And that movie was released a couple of years ago. If you're interested in, my wife, Barbara, is the daughter of the movie maker, and she has the screening rights. So if you want to screen the movie in your club for, I don't know, an evening or kind of for, I don't know, Christmas or so, then contact her, she can organize that for you. So this was all running fine until in 2017, we got a really nice offer from UCR, whether we would be interested to move cyber and the approach of cyber basically to California. And that was quite tempting for us for a couple of reasons. I'll try to quickly kind of summarize like why we kind of decided to move from Western Australia to California. The first one is that UC Riverside has become quite a hot spot for insect research. I don't know whether you're aware of that, but if you look at the global rankings of entomology departments, or departments studying all sorts of insects, UC Riverside actually ranks second in the world. So, and that was, we knew about Riverside before that. So it's quite well known for like the expertise and also the infrastructure that is around. So that was quite attractive for us to go to a place where there's an entire department working on insects, and there are other people working on bees as well. But it's also Riverside itself. Riverside is basically a very interesting spot. It's very close to the Central Valley. You all know that, right? That stretch between Los Angeles and San Francisco, which is like a global red hot spot for the production of a lot of crops. I think it's worth $8 billion each year. And of course, like the most kind of iconic kind of crop that is grown there are the almonds. I think as of today, more than 80, well, I think it's closer to 90% of the global produce of almonds is now produced there. Almonds completely dependent on insect pollination. So in order to achieve that, each year there has to be these 1.2 million beehive transportation into the Central Valley, which is the largest global man-made pollination madness, right? Which happens every year. And just kind of to compare that, in the entire US, you only have these 2.8 million hives left. So it's like close to half or almost half of the managed beehives that are left in the US actually have to travel to California to kind of secure these pollination events. I think what's furthermore really interesting in Riverside, in the southern part of Los Angeles, including Orange, is that like you have this incredible bee diversity. In fact, like Southern California is a red hot spot and globally recognized for its diversity of bees. We have already put up not only one bee hotel, but already several of them. And this year they're just filled up to the roof. It's like amazing to see how many bees are out there and how many bees you can attract with relatively little effort. You get them into your backyard. The third thing that I was really fascinated about Riverside, or we were really fascinated about Riverside, are the Africanized bees. You all know them, right? They're kind of a little lab accident in Brazil. 1957, they escaped, and then they traveled north. They came into the US, I think 1986. that correct? I think it was 1986. Well, I think it was the same year as you got varroa mites into the US. But the Africanized bees basically stopped going further north, I think then something between Los Angeles and Fresno or so. So they're in the southern part of the United States. You have these bees hanging in the trees. They survive without any human support. So for me, these bees basically are very interesting because I feel I can learn a lot from these bees, because they must have the ability to cope with the harsh climates here, to cope with varroa and so on, right? So I think like they can teach me a lot, and California and Riverside would give me the opportunity to actually access them and study them. Now, when I arrived, we quickly kind of became aware at the university there's quite a number of people that are interested in bees and pollination and bee health. And so the center actually grew relatively fast. And these are the people at the moment that, that are involved in the center. So these are the PIs. I'm not going through them one by one. If you're interested in, I can do this. I will kind of mention some of them later on when we talk about what we actually now do within cyber. Now, what's the approach of cyber? We initially had a conference in Perth where we invited the beekeepers, and we basically asked them, what would you need in the next 5 or 10 years that would actually make your beekeeping easier or more profitable or more sustainable? And one of the beekeepers, which was a 4th generation beekeeper, and he was already in his 80s at that time, he had quite an elegant idea, and actually we picked that up and we kept it to today. And that is this toolbox idea. And he said basically that if you have 3 beekeepers in a room, you typically have 5 different opinions, and that's just how the world is. And you have to kind of live with that. And from my interactions with the beekeepers here in California, that seems to be a global phenomenon. So that's what it is, right? But that's fine. What this beekeeper basically said is like, the ideal thing for us would be if you develop new tools. So a beekeeper has a toolbox. You have a smoker in there and a bee brush or whatever you have in there. You have some management tools in there. If we develop new tools, it's really up to the beekeepers whether they want to take it up or not. If it's a successful tool, it will spread. If it's an unpractical tool, it will not be taken up by the beekeeping industry. So we thought that's actually a cool idea, and we started to develop ideas with the beekeepers of what type of tools might be really interesting or really important. And we came up with an approach that I show here, where at the bottom here, we have 2 approaches that science basically takes. We call them systems biology and behavioral ecology. Now, systems biology are people that are really interested in individual substances and molecules. There could be a gene, there could be something that the bee produces inside her body, right? So systems biologists are normally kind of chemists and biochemists and geneticists and people, these type of people. Whereas like behavioral ecologists, that's more like where I came from. So these people go out and they do experiments in the field, and they study bee health and performance in the field. And we basically kind of decide like there are 3 major outlets that might be quite interesting to develop further. One is like to study the reproductive biology of bees, which will really be helpful if you, for example, are interested in bee breeding of healthier bees. We know that like bee diseases play a major role, and that's again like a global phenomenon. So we should kind of look what type of new tools or medication we can develop to kind of control them. And we should probably start to understand the bee better in its environment that it performs, right? Which is a very different one in California than it is in Washington or Maine or in Europe or in Australia, right? And with the idea that like, if we understand that interaction, if we could monitor the health of the bees, that would give us much better kind of tools to recognize early problems and then kind of be better able to kind of deal with them. So with this is like, we started that idea with breeding for more healthy bees. And as you might remember, I did that experiment at the start of my PhD, right? Where I developed an artificial insemination technique for bumblebees. And I made a very interesting observation during that time. So here's a picture that I took during my PhD of a bumblebee ejaculate. And that basically looks pretty similar to a honeybee ejaculate, right? So on the top here, you have in white, you have the semen. And then on the bottom here, you have a huge part that we call seminal fluid. And that's a sticky secretion. And when I saw this the first time, and if you have seen a honeybee ejaculate or an ejaculated kind of honeybee male, you might also have wondered, so why is there all this kind of sticky, gooey substance down there? What is that substance doing? Why is it there? And in the bumblebees, I asked myself the same question, and we started to study this. And what we found is that when you transfer that seminal fluid to the female, she stops mating. So she becomes hostile towards males. And if you give that enough time, the female even becomes aggressive towards males, and bumblebee queens that stung males to death. So, and we started to identify like the components that are in there, the substances that are in there that are responsible for that. I found that quite a remarkable effect of that seminal fluid. So given how much seminal fluid we have, I started to kind of define this seminal fluid as a magic liquid, and I wanted to know what's in there and how does it actually function. And to do that, we basically did not use bumblebees. We switched over to honeybees for some technical reasons. And I don't know whether you have seen that before, but that is the sexual tract of a honeybee male. And it consists of two parts. You have these glands here, these accessory glands, and they produce the seminal fluid. And you have these accessory testes here, and that's where the mature sperm is stored. Now, what you can do is you can dissect these males, you can sample this seminal fluid, and you can then start to use biochemical technologies to see what's in there. And the first way to look at what's in the seminal fluid is to put that on a gel and to separate that. So we colored all the substances that are in there in blue, and then we separated them with electrophoresis. So you have these dots here, you have bigger and smaller dots, and each of these dots is one of these substances. And I refer to these substances as my little James Bonds, okay? Because all of these substances have some function and they do something. Although at that point, of course, we didn't know what that would be, but you can already see that there are like really big dots. So for some of these substances, there's quite a lot of around. So maybe they are really important. And others, there are very minute little kind of substance amounts around of these things. So what modern research can do for you is you can actually take such these James Bonds, you can cut them out, and you can use like a gigantic microscope. We call that a mass spectrometer, but the idea is basically you focus into that single spot and you can identify what it is. And then you have a name of what it is. It's a name of a protein, and we got like lists of hundreds of these substances together, and that allowed us to predict of what the type of functions you would see in there. And we came up with 3 different functions. We had like James Bonds in there, and they keep the sperm alive. And that's probably what you would expect, right? In seminal fluid, that it helps the sperm to survive during the transfer to the spermatheca. But then we had like things that we were a bit surprised about. One group of molecules that we met were involved in some warfare. So these are snipers and agents that are not nice to each other or to others. And we wondered like what they do and why they are present in the honeybee ejaculate. And then we had like a group of agents, and they basically are part of the honeybee immune system. So if we look at the seminal fluid, it's basically like a Swiss Army knife, right? So it's like one liquid, but it has multiple functions. But the really cool thing is that like we can collect it from bees, we have it in a vial as a liquid on our bench, and we can do experiments in the lab, and we can play around with this and understand of what it can do, how this changes between beehives, how it changes between bee races, how it kind of changes based on the genetic of bees. So we kind of wanted to know, like, what can we find out of what's happening in here? And the first thing that we did is, like, we wanted to know how effective is the seminal fluid to keep sperm alive. And you have here a graph where we exposed semen, the sperm cells, to seminal fluid, and you can see almost 100% here after a couple of hours are still alive. Whereas if you keep this sperm without the seminal fluid, they start to die off. So at that point, only about 50% is alive. And the remarkable thing is that you can dilute the honeybee seminal fluid down 100+ times, and it still has the full capacity to keep the sperm alive like that. And we started to become quite interested in, like, what the abilities, or what our abilities could be if we know the agents that are responsible to that, to start to store sperm over longer periods of time, which would be actually quite interesting. You might have heard that people try to deep freeze bee sperm, but that is still quite tricky to do and not so straightforward as, for example, in invertebrates. So then we had the second kind of group of proteins that are naughty ones, nasty ones, not something that you would upfront expect there. And like, so we did some experiments to kind of get further into that. And the way we did that is that we used drones or males from different hives. We dissected their sexual tracts, and we collected the seminal fluid from a male from one colony and the sperm from that colony, so from that male. So we have a focal male and we have a potential competitor. And now what we do is, like, we expose that sperm to the seminal fluid either of the same male or of that potential competitor. And then we are interested in how happy that sperm is. So how do you measure sperm happiness? Well, we have two dyes, and they distinguish between happy and unhappy sperm. So here, what you can see here is the dyed sperm. And the green sperm heads here, they are happy sperm because they're alive, and the unhappy sperm here, they are red because they're dead, right? So they're killed. So if you do this type of experiments, what you can see is that the males use these seminal fluid agents to kill the sperm of their rival males. So if you have a queen flying out, mating with multiple males, there is carnage inside the sexual tract of the queen, because there is immense warfare going on inside the queen's sexual tract, because the males try to kill each other's sperm because nobody wants to share paternity. The interesting thing is that we, with these biochemical kind of approaches, we have started to learn to stop this. So if we, for example, think about long-term sperm storage, right, this would kind of clearly have a negative effect if we just leave the seminal fluid on the bench for long periods of time. We can stop this, and there would be, again, like a way forward if we think about whether we could kind of preserve sperm or specific bee genotypes in the future. Now, this can't go on forever, because the queen bee, of course, needs that sperm to fertilize eggs. And I assume you all know that, like, queens never remate, right? So they have a mating flight early in life, then they store the sperm, they will never replenish that. So we studied the queen side of this as well, and what we found is that the queen actually has her own agents, and the own agents go in and specifically kill those agents that are responsible for the sperm killing. So if this is a bit confusing, I can summarize that in a single picture that you can see here. So this is what we think is happening. You have the female that provides the sexual tract. So that's the colosseum here. And then you have the males, and they enter as ejaculates, and they carry weapons, right? These agents that I've talked about. And then you have complete losers, like the one here in the background. You have winners, and then you have others, and they are kind of left at the mercy of the female up here, right? You might know this very famous sign that they made in the ancient Rome. So if the thumbs are down, he has to die. If the thumbs are up, he can survive, right? So in this case, like, the males fight over paternity, but the females basically rule the world, right? At least the bees. And I think that has really interesting ramifications if we think about artificial insemination, because to a certain degree that helps or does not help us, especially when we're interested in genetic diversity or when we're interested in breeding specific lineages or lines, right? Because this dynamic inside the female will always bias paternities towards some males. But there was another effect. When we looked more carefully at these agents, there was a second James Bond in there, and that James Bond was very targeted to attack the neuronal system of the queen bee. So we looked at that. So that's a scan, a CT scan through one of our honeybee queens, and you can see here in the middle, that's where the brain is. And we artificially injected seminal fluid into queens, and we looked then of what is happening inside her brain. So what's the activity in her brain? And what we found is that the activity changed quite dramatically, and those genes that are responsible for the visual perception of the queen, they're turned off. So we hypothesized that the male bee, the drone, through the seminal fluid modifies the visual perception of the queen. And we found a way to test that. We built this little kind of device here. So this is a worker, but we also did this with queens, where you have a worker in that device, and then you have a little diode here, and that diode flickers light. And we can measure the bee's response to that light. So how well is the bee able to distinguish two flickers from one, and how long do you have to kind of provide the bee with a light impulse until the bee reacts to it. And what we find is like that as soon as the queens receive seminal fluid, over time, a day or two later, they actually lose that capability. So they cannot see that well anymore. Well, with other words, it's like, indeed, like the males seem to blind the females by love. And that would make sense if they can actually avoid that the females go out and mate again. It's a bit negative effect on the beekeeper side, because if the bees don't see that well anymore, there's a risk that they could not find home, right? And we tested these ideas because the beekeepers were quite interested in that. And that was work that was done by my wife, Barbara. So I can only tell you the details. She should basically give this talk about her work here. What you can see here is a queen bee, and Barbara glued a little RFID tag on the back of the queen, and that allows us to trace the mating flight behaviors of the queen. So we know when she's going out, and we know when she's coming back in. And what Barbara found is that the drones manipulate the queens, and if the queens receive the seminal fluid, they fly out earlier to avoid these, that they have lost their visual perception. If they go out later, the chances that they don't find back or that they don't return to the hive actually go up. So this is like, so these male effects have actually quite some impact on the performance of your queens and the mating success. And that's important because, as I said before, queens never remate later in life, but they still are able to kind of store all that sperm throughout the rest of their life. Not in California, but in other parts of the world, they can do that for up to 7 years. And one of the questions that we got from beekeepers was that, like, how long can a queen actually survive? How long is she able to lay eggs? Or when, with other words, when do you need to requeen a hive? Because your resident queen is starting to lose her fertility. And to kind of get a bit into that, we found a technique where we can count the number of sperm on each of the eggs that the queen lays. And that looks a bit like this here. So we have here an egg, and then we have here again that dyed sperm head of a honeybee. And then in the center part here, we have something that is referred to as the micropule. The micropule is like where the sperm has to swim towards, and then this is where the fertilization takes place. Now, if you do this for a couple of thousand eggs, what you will find is that queens are extremely economic in their sperm use. They only use about 2 sperm per egg. And because we know that they store something around 4 to 5 million sperm at the beginning of their life, they can sire something between 1.5 and 1.7 million offspring, right? So it's like, this is quite remarkable. If you know how your queens lay eggs over the year, you can actually predict when a fully fertilized queen is starting to run out of semen. And beekeepers in Australia, for example, used that, and they extended their requeening, because based on this data here, the queens that they when they wanted to requeen them, they still had enough sperm in their spermatheca. And that's just like the incredible small amount, that's like thousands of a volume of a needle pin, what the queen takes out of the spermatheca for each fertilization. And as you might know, she's doing that she can do that up to 2,000 times per day, which really kind of, I find like absolutely mind-blowing of how they can do that over an entire lifetime. All right, so that was on the warfare side, and then we had this kind of third kind of group of agents that we found, which are linked to immune defense. And the interesting thing is that, like, what we found in terms of the immune defense inside the bee. And to do this, I have to show you, unfortunately, a really complicated graph, which is this here, this figure. It is not as complicated as it looks like. So here at the top, we have the baddies, right, that arrive. So there could be fungi like Nosema. It could be injury or stress when a varroa mite comes along and drills a hole into our bees. Could be bacteria like American foul brood or European foul brood. So here are the baddies. And what you have then in the bee immune system is some recognition systems here. And I call them James Bonds. Here are the real names of those. I don't know why people came up with that. But these are basically these agents, and they are able to recognize these baddies. And they then transfer that information inside the bee here, and then the bee responds to that with an immune response. Now, the interesting thing is, like, what of this did we find inside the seminal fluid? And the dot or the stars here, they basically indicate those agents that we identified. And what you can see is that almost the entire immune system that the bee has available is present in the seminal fluid. And I think that's really interesting, because again, we can sample seminal fluid and we can study the functioning of the immune system on the benches in our labs, right? And that is much easier than if you have to do it inside the bee. The big question, of course, is why would honeybees do that? And I basically had the suspicion that, like, there must be some baddies around here in the ejaculate. So I punished one of my poor PhD students. I said, it must be somewhere, just find it for me. And it took a while and some tears, but she eventually came to my office and she showed me this picture here. And you can see here this string, so that's the semen. And then what you can also see is these little kind of bubbles here. And that's Nosema apis, but we also found later on that Nosema serrane as well is present in the ejaculate. So these both fungal diseases that are really kind of widespread globally, they're also here in California, certainly Nosema serrane, they are able to contaminate the ejaculate. And that's a problem, right? Because if we do artificial insemination, if we transport semen around, that means that there's always the risk that you have uninvited guests in here. And Nosema is not the only one. We also know that viruses, like the deformed wing viruses, so they can also travel in that semen. So artificial insemination is a wonderful tool, but you also have to be careful of how you use it, because there are also some dangers to that. But what we had is, like, we found this immune system, we found a parasite. And because we can do everything in the lab on the bench, we started to kind of have little war games taking place on the bench, right? So we collected seminal fluid, and we collected the Nosema spores, these fungal spores, we put them together, and we wanted to know what is the effect. And the effect is shown here. So you have here the parasites that are just kind of in a normal kind of liquid kept on the bench, and you can see 100% or close to 100% of these parasites are just happily surviving in there. As soon as you add seminal fluid over a time period of 5 to 10 minutes, almost 80+% of the spores are almost instantly killed by the seminal fluid. So that's incredibly potent. These immune agents inside the seminal fluid are remarkably potent to kind of kill these spores off. And we looked at that under the microscope, and that was quite interesting because you can actually see the spores dying. There's nothing more satisfying than to see kind of bee diseases dying off under your microscope. And what we saw is there are actually two different ways of how the bees kill these spores. One on this hand here is just a straight killing. And we know now 3 substances, or we have the names of 3 agencies that seem to be responsible to do that. And we have a PhD student at the moment that was supposed to test them in Riverside of whether we can actually confirm that we can use them as potential future medication. And then you have like another agent here, and he is doing something different. So what that substance is doing, it sends a wrong signal to the parasite. So these fungal spores, they multiply in the midgut of the bee. So they wait of a signal that simulates like, okay, you're in the midgut now. And then the spores have a little string that comes out, and they look for a host cell in the bee gut and penetrate that, and then they deliver the DNA of the parasite into that cell, and then they multiply, the gut cell bursts open and releases more spores. Now this agent here is able to mimic that. So it sends a signal to the spore, you're in the midgut now. The spores send that little string out, and we can find then the DNA here in that liquid, which is seminal fluid, but they can of course do no harm. So it's quite a cunning way to trick the parasite and to kill it. We also try to see whether the seminal fluid is active against other other microbes, for example, yeast or things like that, and we never found anything. We find that the seminal fluid is only really active against these Nosema spores. So we have like a high level of specificity. The spores are killed with accuracy, they are recognized and killed with accuracy, and the redundancy, so you have 2 different ways of how the spores can be killed. And I think that's really cool because that was the first time that I saw substances that are derived from the bee's immune system and they actively killed a disease. In this case, it was Nosema. Right? And as I said, at the moment we have 4 or 5 active substances that we have developed, and I think there's a lot of kind of opportunity here, either to develop new medications or to look for bees that have higher levels of these active substances and then use them as new breeding stock. And because that worked so nicely in Nosema, we started to kind of be interested in whether we could see the bee immune system working in other diseases as well, and we decided to go for Varroa. So we exposed Varroa mites to larvae, because in the end, Varroa is a larval disease. And we did that in 3 different bee species. We did this in Asian bees, which are the original host of the bee, and they actually cope with the mite. And we did it in 2 honeybees, the African bee, not the Africanized bee, but the African bee in Africa, and the European honeybee, so what we have around here. And the interesting thing is that the Africanized bee is, of course, a mixture between the African bee and the European bee. Right? So these are parents of those. What we see is that the Asian bee has a full set of immune genes that they upregulate when the mite is coming close, not when the mite is biting the larvae, but the larvae recognizes the presence of the mite and triggers immune response. And we can see some of these responses also in the African bee and in the European bee, but it's not a full set. Interestingly, the sets are different in the African bee and the European bee, and if you combine them in the right way, you actually get very close to the Asian bee. So the Africanized bee could have been lucky to get the right combination that basically allows her to be more tolerant against Varroa mites. And one of the big things that I would really like to do is now to repeat this type of experiments on Varroa using Africanized bees, just to see whether we are on the right track, whether we have identified the right agents in there that are responsible for that. So we are interested in kind of studying what I would call feral bees or survivor bees, and to see whether we can see these traits, or we can confirm these traits in the local bees here as well. That I'm convinced that they're able in one or the other way to cope with these mites. So far I've talked about with the reproduction in a way like what we have done here, and how we can use these for further breeding kind of approaches. And I also already mentioned like how we started to kind of develop new possibilities to treat bees in the future. I think what I'm really excited about here is that like we develop substances that are part of the bee. So we are not kind of coming with something that is not natural in the sense that like it's a product that would not occur in the beehive. The substances that we identify are naturally inside the bee and are naturally there as part of the immune system. And that of course would solve a lot of problems, because like, first of all, it might be much easier to get that regulated if you say, well, it's just a naturally occurring bee product. And secondly, you don't have like the problems of spillovers into honey, right? And the problem that like residuals of chemical treatments in honey then resulting in either kind of the decline of the honeybee price, or kind of that you can't sell these kind of products anymore. So the last thing that I quickly want to mention is, so what are we doing here on this health monitoring effect? And initially I thought like we should really kind of hook up with the veterinarian sciences, because veterinarians are so good at like treating every domesticated animal that we have, right? If you think about dogs or pigs or sheep or whatever, the veterinarian sciences are really good at like recognizing what's wrong with them. They have hundreds of medications available for these animals. So we are really good at keeping our other domesticated animals around us really healthy. But the vets were not really interested in bees, and they were not really interested in me. So I had to come up with another idea. And the other idea that we have started to cook up here, or that we continue to kind of really develop at UCR, is that you have your little beehive, and your beehive is happy, right? Now, how do you measure happiness in a beehive? Well, we know that like bees, the language of bees is sound and smell based in the end. So if you have the right type of sensors in there that basically continuously measure these kind of parts, we could sniff in, or we could listen into these hives, and as soon as something bad's coming along, right, we would smell, or we would hear a change in the behavior of the hive. The sensor would then kind of recognize that, send an alarm to your mobile phone, and in the ideal case, it will actually recognize what the problem is, right? And it would alert you of what's wrong with that hive. So it's not like necessarily easy to do. What you certainly need is, apart from building the sensors and making that work so that the bees actually accept these sensors inside the hives, you also have to make these sensors clever, in the sense that like, we don't really have the time to download like every second day all the data and then do statistics on that. So the sensor must be able to do an analysis of what he measures and what that means by itself, right? And so we have started to add electrical engineers that build these setups. We have started to add engineers from that work on artificial intelligence and machine learning. And we have added a lot of biologists that basically can deploy these kind of sensors into beehives, and that can also kind of identify how bees respond, for example, in differences in smell when there's a specific problem coming along. And so, although this might all sound like a little bit like Star Wars, we have now the third generation of our sensors that we deploy, that we have deployed, and we basically kind of continuously circle that around. So we have these honeybees and these honeybee hives in our apiaries, and we stress them, or we kind of expose them to certain environmental stresses, and we test how these bees respond, what type of chemicals they release. We also have some chemicals that are already known from the literature. Right? We know there's a hunger signal. We know there's a signal that triggers hygienic behavior. We know the queen pheromone and so on. Right? So we have this. We provide, we can identify new ones using these kind of approaches that I've shown you before with this mass spectrometry. Once we have the names of them, or we have these substances, we hand these substances over to our engineers. They use existing sensors, or they use new sensors that they build to basically chime into these molecules that we're interested in. So you can either have an existing sensor that you can train to smell what you're interested in, or you build a new sensor, which is much more work, but depending on what's on stake, it is actually really interesting to do. Once they have that, we integrate that into the platform that we already have, and we kind of deploy it into the beehives that we keep. We collect the data, and we then need like this data analysis people to kind of show us that like the type of data that we collect, that they have some analytical power, right? And we can just continuously do that, right? So we want to add more sensors, and we want to become more and more sophisticated in how to do that. So that's the third part here, where we have started to kind of try to continuously monitor bee health under field conditions. So one of the big things that I've told you about cyber from Perth was like that we really kind of focused on building and maintaining really strong and tight links with the bee industry. And we have started to do this here as well. So I've started to kind of talk to the beekeepers in the surroundings. I'm really happy to give talks to beekeepers. And I think there's a lot of opportunities to do stuff together, right? So I see cyber not so much as a platform or as a structure that is inside the University of Riverside or UC Riverside. I see that more as a link where we can invite beekeepers and researchers, and we provide them with a platform where we can interact and basically do what we have done over the last 15 years. And I think at the moment, this has specific relevance, right? Especially like the value of agriculture, the value of food availability, because for a few weeks now, it's probably something that very few people have experienced, that all of a sudden the shelves are empty, right? And that people have realized that food cannot be taken for granted. And especially affordable food. So I think that's one of the food banks now in Sacramento. So the number of people that cannot afford food, or the availability of food, is something in that entire pandemic that I think has really become a major issue. And the bees, I think, like play a major part in that. Bees for me mean food and also mean income, right? And it is interesting that like if you go to Google, you have a little tool there where you can see what people actually looked up over the past years or months or whatever. So I kind of typed in how to make honey into Google Trends. And this is the graph that I got. And it basically starts at the last recession, and it goes to today or yesterday when I took that graph. And what you can see is that there's a very strong peak that like with like the onset of COVID and the lockdowns and these problems with food availability, people start to become really interested in where actually honey comes from, and maybe also like how to keep bees, right? So what we have started to do is like to look for partnerships with beekeepers in the surroundings, because I'm a strong advocate that like I feel the problems or the issues with bees are most likely resolved on a local level rather than a global level. And what was mentioned at the beginning is that we submitted a proposal to set up a bee breeding program. And we could use the expertise that is already, that we have already, the experience that we got in Australia with setting something like this up. But here, for example, that's an example from Germany, from a German beekeeper school where I was working for a short while, where the beekeeper school maintains some breeding stock, and the beekeepers then come in with their virgin queens here. So each of these boxes is basically little like mating nook, and with a virgin queen, the school provides the males, the bee keepers come along with their virgin queens. They then mate, and they basically take the genetics of that breeding program as stored sperm inside their queens back to their home. So it's a really, I think it's a really nice way to distribute breeding stock to beekeepers. And I think what we have in mind for this proposal is that there is like quite some observations out there, and we see that in our bee yard as well, is that like we have breeding stock, we buy queens, relatively soon we see like the turnover or requeening events, and then we have like we have colonies that perform actually really nicely. So with every requeening event, we see that like these bees seem to be able to much better cope, especially with diseases. Some of them become really defensive, so that's not what we want, but because they become Africanized, but others seem to be able to, we seem to be able to really nicely manage. And I think that's the type of bees that we're really after, right? So bees that have a good temper, but that where we actually would not have to treat or not to treat continuously as what we do at the moment. And I think that's like where we are really interested in collaborating with beekeepers, because you all have these bees, right? And you might see these bees. At the moment, it's really difficult to keep them together, because as soon as you lose the queen, as soon as you have other requeening event, they will mate with other males, they will become more Africanized and so on. With our setup, where we can do artificial insemination, where we use this type of island approach, we could actually start to bring that together and keep it together, right? And close it off so that you don't have the effect from outside. And with the technologies that we have, we can even understand the mechanisms that drive the health in these bees. What we have also kind of submitted is a proposal to organize a bee health conference in Riverside in 2021. We sent that to the USDA, and that was accepted. So the USDA basically has invited us to submit like a little bit of a longer summary of what we are after, and we can get money for this. So the idea is to organize that conference, hopefully next year, if you're all allowed to go out again. And the prime idea would be to really kind of invite people like you from the surroundings, beekeepers from the surroundings, and to isolate or to identify, so what are the major obstacles that are in the way here? What would we need, or what would you need in 5 or 10 years to have like sustainable bees, to have healthy bees here, and then to see like how that can be translated in different research projects. And we have, bees are kind of in fashion at the moment, so it's actually quite easy to recruit students at the moment to do this type of research. And I think bees in particular, because it's not so much academic, I think students really enjoy if they work on a problem, on a scientific problem, but they can see that it actually has an applied effect, or that there is some relevance out there. What we also have is thought, or start to discuss, is like whether it would be helpful to have some form of, well, I call it here beekeeper school, but more like a place here in the southern part of California where we could offer some classes or courses in areas that are of interest. There could be artificial insemination or grafting. I had very long chats with Elina Nino from Davis, and she's very supportive of this because she's just overwhelmed with the amount of work that she has to do. She's responsible for entire California, and I think that's just a really, really big state for one person. So she's really supportive to build something similar to what they run in Davis here in the southern part of California, and we have some other kind of institutes that have become interested in it. So Cal Poly Pomona, for example, is really interested to join such an approach, and also UC San Diego, where we have some beekeepers as well. What our advantage is, like, we teach bees in beekeeping at UCR. It's a class really high demand. We normally get like 4 times more students that want to join the class than we can offer places. So we offer it twice a year now. We are running at full capacity. But we have all this set up, right? So we have a teaching apiary with about 25 hives. We have, I think, 7 or 8 artificial insemination equipments. We have the lab. So we can actually offer the beekeeping side. We can also offer like the infrastructure to do this type of activities. If you're interested in what we do, we have a web page now at UC Riverside where like the different kind of PIs are presented. There's some information about what we are after. There's also a Facebook page and Twitter page linked. So that's where we normally, when we publish something or when there's something coming out, we place that there. That has been very silent over the last couple of weeks because we're basically just shut down, right? So there's nothing happening at the moment. But here's the website of Cyber, which has these links to Twitter and to Facebook. And I think with that, I'm done, and I'm happy to have a look at your questions if you have any.
Awesome. Well, thank you so much, Boris, for all that information. Yeah, there were a couple questions that came up, and I wanted to go ahead and open it up. If anybody else has additional questions, please feel free to type those into the chat.
Rick, Boris,
yeah.
Can you put your website back up there so people can copy it?
I put it in the chat. Is that okay?
Yeah, that's great.
Okay.
Yeah, and from the chat, for everybody that's here listening in, from the chat, you can copy and paste that text on your machines, so you can open that up in your own web browser as well.
To be honest, if you Google my name and Cyber, it immediately pops up. But I put the website in for you.
Awesome, thank you. So if you have questions, please feel free to put those in the chat box or be ready to raise your hand. Again, if you haven't used Zoom much, down at the bottom where there's the participants button, that will give you some icons, including the raise your hand. Boris, I saw that website pop up. Thank you so much. cyber.ucr.edu.
I have to send it to everyone. Here we go.
Well, there it goes. Perfect. Sorry.
Yeah.
Yeah, one question actually was regarding cyber. Is it possible for people from, quote unquote, the outside of UCR to be involved in that?
Absolutely. I think that's the entire idea behind it. So
in what ways and how?
So I think so I gave talks at the LA Beekeepers at the I think we have 4 beekeeping association or clubs in the surroundings. I've talked to all of them. I invited all of them to join this. What would be ideal from our end or from my end is that we can offer from our end that if we have activities, if we have collaborative research, if we plan things like that, that we keep you informed. With the other bee clubs, I basically say, if you're interested in that, can you nominate somebody on your end, which is like, I don't know, a communications person. And I keep in contact with that person and basically say, okay, this is what's happening at the moment. And there will be things happening, right? So if, for example, if we get invited for this pre-breeding program, this is like a pre-proposal, so you have to kind of line out your ideas. I don't know how many pages that was. If they like our project, you still have to write a full project together, right? And I think at that point, it would be really interesting to get the feedback from the beekeeping side. So that from the writing of the project, that we can build that link and we really factor that in, right? If we start to organize that conference, I think that's another kind of thing that you might be interested in to join. We're also interested in feedback on that. We need to write the grant. We haven't done that just because with all the teaching and all the other kind of weird things happening in our lives, is like we haven't done that, but that's what we're going to do over the next couple of weeks or so, that we write up this. If you have any suggestions of for example, what we should discuss there, what is major topics of interest, for example, like what I got from the last talk is that there is major problems with legislators, so that each city and each county has different kind of regulations about who is able to allowed to keep bees and how many and things like that. So there was something brought up. So if you think about a conference that kind of defines a 5 or 10 year roadmap, if you want to get something covered during these 2 days, let us know, and we see that we can factor that in. And then, like, as we move ahead, I think like what we should be really good at is, like, as researchers, we apply for money, we can generate money, right? So we can basically write grants, we have access to to these grant funding agencies, so that we can generate money to do certain activities. And as soon as that is happening, I think would be very interesting. For example, for a breeding program, to have people that would be interested to join this, either because they have bees that they feel are really interesting and need very little kind of maintenance, or to test certain things. So once we have these sensors, for example, we have only 80 hives, right? So at some point we get to the limits of what we can do here. But if there would be some people on your end interested to try them out under realistic conditions, as a backyard beekeeper or semi-professional, I don't really care that much, right? There is something that I think like we could start to build these connections. Does that help?
That's great. Thank you so much. And Liz did mention in the chat, I don't know if you wanted to elaborate any more, Liz, that the club will be involved.
Yes, we did submit a letter of support to Boris and the group, the cyber group, in preparation for their grants. So once things are solidified on cyber's end and whatever help they need from beekeepers, at that point in time, we will be notifying everybody. So if they're looking for very docile hives, you know, colonies, or if they're looking for specific items, we will put that out to the club as a whole. And then you guys can respond back, and, you know, we'll work out the details later, but this is a community, you know, experiment and everything that's going on, research project. And like Boris said, LA Club, Long Beach, I don't know about Basque, but on this side, and I'm not sure about San Diego, but I'm sure you guys have approached them as well. And like Boris said, because all of our areas are so diverse, even if we're like a mile apart from each other, I mean, you could have the ocean breeze and then you could be, you know, a couple miles inland. So there are going to be a lot of things that they're going to be asking for, and we are here to help, and they know that. So if that answers anybody's questions on the participation, if you have any other questions about that, you can always email us, or you can email Boris directly. And yeah, so any other questions from anybody? Pamela.
Thank you, Liz. Yeah, Pamela, I'll get you here. Go ahead.
Thank you. Currently, where are you getting most of your funding for cyber, Boris?
At the moment, so when I arrived here, the UCR gave me a very nice startup. So that's most of the funding is coming out of that startup that runs for 6 years. Okay, we have now, I think, 2 years something we have used up of that, but this is running for at least another 3 years, right? So we are set with that. And then, like, of course, we apply for grants, right? So we apply for research grants that can be either very, very academic, and then we send it maybe to the NSF, National Science Foundation. But I think especially for bees, there is a lot of opportunities to generate funding for the type of activities that we have in mind, right? Project Apis M. We have within UC, we have a lot of opportunities, and I think at the moment, especially bees and agriculture could have a really nice go, because I get some flood of emails now that, like, there will be money made available to basically get the economy up and running, get the agriculture up and running, to create jobs, right? Like, you have the most brutal recession, I think, since the United States is in existence, right? lots of people have lost their jobs. So as I said, in my view, beekeeping is system relevant. It's an art to do it. You know that. I had to learn this the hard way, especially in California. It's like unbelievable. That's how we keep a colony alive here, right? Compared to what we were used in Australia. So there's all this expertise around. I think like in the long term, you know, thinking about what would be a profitable, sustainable beekeeping industry here. And that doesn't mean that it's an industry, but it could be that like, I include in my thinking, I include the hobbyists into that because they basically also safeguard in the end against like complete losses of crops, right? So they could guarantee pollination in the future. And so I think like we have these opportunities. I see them coming. So it's not yet clear how that works, but I see that there could be opportunities to apply for funding. So if you get some feedback from your ends, right, we can start to kind of turn that into proposal. We can turn that into research projects, and we can apply for that funding. Is that helpful?
Yes. Yeah, and I wanted to comment also that I thought your presentation was excellent.
So,
I do. You made a comment about the movie More Than Honey at the very beginning, and you had mentioned that you were considering putting a documentary together to dispute it or support it. I was a little confused there.
okay, so do you know the Bee movie? There was a cartoon that came out 2008. Don't worry about it. It's I don't know what it is, but it was a movie that was supposed to bring up the bee problematic, but it really failed on the entire. So we were so miffed about it that we forced our father-in-law or my father-in-law to do another documentary that in our view depicts the bee as it is with the right biology, and also kind of provide some insights into how dependent we are on bees and also the relationship between humans and bees. That movie is called More Than Honey, and that's already in existence.
Yeah, I saw that movie at the Palm Springs Film Festival about 4 years ago. It was excellent. That's why I was a little confused. He did a great job.
Okay, I will forward that. It was interesting to film that because I'm a scientist, so there are numbers and figures and that's it, right? And then you have like a movie maker and he wants to tell a story, and he packaged that up. So it's a scientist fighting with a movie maker. He's like, no, you can't do that. That's not how it's done. But
no, it was so informative and very engaging. Yeah. Anyway, thank you.
As I said, we can show you the movie. We are not charging you for that. So we have the rights that we can show it for educational purposes. And that's what we do. So
I encourage anyone who hasn't seen it to download it and view it. It's great. Thank you again.
Thanks, Pamela. I appreciate that question. And Boris, awesome. Sean, I see your hand. Go ahead. I'm trying. There you go.
Okay, there we go. I was curious, Boris, when you do your artificial insemination, are you using drones from multiple hives so you get genetic diversity? Or are you using drones from a single hive, which are thereabouts all twins, to get a particular outcome?
That depends what I'm after. So if I'm after, let's say, stock that I want to keep, for example, the teaching yard, we always use a mixture. If we want to show a certain trait, or we want to isolate, like, okay, this is like, we want to identify a specific agent and show, okay, that's the James Bond responsible for that. We have tried single drone insemination. We have also done like inseminations with drones from the same hive. I think so for experimental or for research purpose, we do it. But any bee that I actually need in a longer term, I always look that I have that I keep the genetic diversity in the hive, because I think we have very good data now that's really a driver of bee health.
Okay. do you find that if you extract the semen from drones of all one hive, being that they're all twins, are the sperm happy? Because, or does it vary from drone to drone where the seminal fluid from the drones of a same hive will kill the drones of the other sperm of the same hive?
Yeah, that's a really good question, because initially I thought, well, the brothers are not going to kill each other's sperm, huh? Why should they? And so we tried it, and the brothers kill each other's sperm. So don't ask me why, they just do it. I think it was less, but interestingly, they can do it. I was really puzzled about that, because we have an immune system and we have specific molecules, and they really learn. They're very sophisticated, right? So when you get vaccinated, you train some of these agents to recognize the disease, and you have a memory. And honeybees don't have that sophisticated type of immune system. So how on earth they are able to recognize the sperm from a different male, or even the sperm from a brother? That's quite remarkable. And they are not the drones. They are coming from unfertilized eggs of the queen. Huh? So technically they're clones, right? Because there's no father. They're all like variants of the same queen genotype.
Which is why I would have thought that they would have had the same agent in their seminal fluid, and thus that wouldn't kill each other, and you could possibly store up a whole lot of semen just from all of those drones from a single hive, and then mix it later on if you wanted genetic diversity.
I'm with you. I think like what saves us is that the queen is diploid, right? So she has two sets of genes, and the male only has one. So as soon as you split that off, right, that means that like let's say the queen has the killer agent, so she has two genes of the killer agents, and depending on what the male gets, could be getting either the one or the other. So that could be the reason why they still fight.
Okay.
But yeah, I agree. It's like it would have been really nice if they don't do it. If you think about sperm preservation or a sperm bank or something like that, which could be really helpful in the long term. And there are some activities in the US now. You might have heard about that one of your top shots in the artificial insemination world, which is called Sue Kobe. She has actually traveled all around the world and sampled bee genetics, and has started to cryosperm, to cryopreserve them, so to keep them at really low temperatures. It kind of works, but is by far not as efficient from what we can do for vertebrate sperm. So there's still a lot of improvement that could be done.
Okay. Another question. You said that you, down in Australia, they were getting upwards of 650 pounds of honey per hive.
Yeah.
Are they putting them on crops, or is this on natural vegetation? What are they? Are they migratory beekeepers?
The migratory beekeepers, because you don't have varroa and all the diseases, and certainly not colony collapse, you have a huge population of feral bees, and they're doing all the pollination. So in Western Australia, there is no paid pollination. So what the beekeepers do is they basically chase the honey flows in the national park. So Western Australia is a gigantic country. It's, I think, 6 or 7 times larger than California, and has 1.7 million inhabitants. You have what, 30 million or something like that, huh? So it's a country that has one city, which is called Perth, and that's where everyone lives, and the rest is empty. So the beekeepers go out, and they are in these pristine landscapes, and they go for the honey flows of these eucalypt trees, and the bees are just happy, so they just keep them out there. They travel from, it's the other side of the world, right? So winter and summer is the opposite. So they go north and wait for the spring to arrive, and then they move the colonies south as like the spring comes further south, and then they have different varieties of honey. And some of their honeys have really high antimicrobial activity, even higher antimicrobial activity than New Zealand, the Manuka honey from New Zealand. So it's a very nice tasting honeys. And yeah, unfortunately they don't get the price for it. I think the price is far off from what Manuka is making at the moment. But I think that was something that we try to support there as well. That is like, if you have healthy bees and you produce honey with healthy bees, that you can actually commercialize that as a green, clean, and healthy product and get the honey price up. I don't know what the honey price here is in California. I assume you're not becoming millionaires by selling your honey. That's just how life is, right? It might change in the future, right? Because I think all of a sudden we have an entire generation of people that have realized that you can't take food for granted. And maybe there's more of a recognition also about like the skills and also the work that goes into that. And that, like, when I'm out and I'm talking to broader public, I always really promote that people buy honey locally and from a local beekeeper to support that beekeeper.
Okay. Yeah, in California, we can sell commercially wholesale for about $2.25 a pound maximum, and then they're selling. If you go to like farmers markets, it's anywhere from $10 to say $14 a pound.
That's more where it should be, right? I mean, the New Zealanders were really clever because they got Varroa and the entire bee industry collapsed. They lost 80% of their hives because of Varroa. And then there was a federally funded program that they set up, and they looked around and said, so how do we make the beekeeping industry profitable, because they need them for the kiwi fruits. And there was this observation about Manuka, but nobody wanted to eat Manuka honey because it's really bitter. It was a waste product, right? So they put some scientists on that, or they looked into like, so what's so specific about Manuka? They found that it has this highly antimicrobial activity. And now if you go to a shop in China or to a shop in Australia at the airport, you buy a pound of Manuka honey for, I think, $480. I mean, I think it's madness, but it just kind of shows like, if you do the right marketing, or if you kind of convince the consumer that this is an authentic product, that the consumer is ready to also invest or pay more for that product. And I think like what you get for your honey, it's just like, it's in the end, I would say it's unsustainable, right? Because like you should actually get more, because you provide much more of a service than just kind of selling that honey. You also provide a service of pollination security and the entire stability of ecosystems is certainly in like agriculture, in urban areas.
Awesome. Thanks, Boris. There was another question that came up in the chat asking about the warfare characteristics. Did you see a difference between European bees and the Africanized bees?
Yeah, that's what I wanted to know, because my prediction is, and I think there were some people that put me on that, because they said like the Africanized bees are much more competitive than these managed bees. So we wanted to do that. It was planned, and then came COVID, right? And they really shut us down. So no one is allowed at the university. We basically sitting ducks now. So I don't know when that will go ahead, but that's something that's clearly on our list, because that really defines as well, like, you know, how much kind of, if you have a requeening event, right, you have a daughter, sister, so you still have the maternal light that comes from a managed or bought queen that you got somewhere. That's what we do. We buy queens. Especially for the students, we cannot have aggressive hives. So we buy queens from the queen breeders. We normally see that they almost, within a very short period of time, they all requeen. That takes maybe months, but typically only weeks. And then you have still a maternal, it's still a daughter of the old queen, but she mates with some bees in the surroundings, right? And it would be really helpful to know whether we get some of the managed bee genetics in there, or whether basically we're just swamped with these Africanized bees that are in the surroundings.
Awesome. Thanks for addressing that question. There was another question that came up that was, there were several pictures regarding sensors and RFIDs, and I saw one in the middle of a frame. Are these things that are just being used at UCR, or are these, somebody said, hey, I'd love to try those out? Is there anything that's more widely available outside of the research at the university?
So the RFID tags where you can monitor bees flying in and out, that is a commercially available system. So you can buy that, right? So we didn't invent that, also because we didn't want to invent it. When it comes down to sensors, there are a couple of companies that now offer sensors so you can buy them. We looked into that. I mean, it's all really starting up. I was not completely convinced about what's available now because the sensors are very often very limited to what they can measure, and they also don't give you a real analysis of what's happening inside the hive. So it's clearly something that's happening at the moment. People build this up. It's like maybe the first mobile phones, right? The first mobile phones that you could phone without some string, and you know, you could hold it in your hand, and it was like super cool. But like 3 meters out of your house and your signal was lost, right? But it was a milestone, right? And I think we have seen that. But with the mobile phones, like what we had at one point was the iPhone, and puff, with the development of the iPhone, you could basically be in any other phone on the globe, right? It basically just wiped everything out. So I think that we haven't seen yet. So I think we need the equivalent of the iPhone for a sensor that measures really specific components inside the hive that we know are relevant, right? Not necessarily CO2 and temperature. They might give you some information, but they don't give you a complete picture of the hive. We want to see the queen pheromone, alarm pheromone, the hunger signal, which is I think oleic acid that the larvae release. So I think we have the knowledge to do that. We just have to put it together and to test it. So that's something that is moving at the moment. And that's why like we feel like there is like that gap for the sensor development. And the companies are not, I talked to them, I went to a couple of conferences where they tried to sell that. It's very clear that like they all would like to have this, but the investment costs are really high. So for a private company, this is not really, you invest so much and you have no idea whether it actually works in the end. That's really in the realm of the researchers, right? That's what we do, that we can generate ideas, we can generate the money, then we do it, but there is no, we don't have the commercial pressure. We are not a commercial entity, right? And so Cyber will not earn money with this or with the queen bees, because that's not what UCR is. We are not interested on the commercial side of things. And that's, I think, where we can provide the research and the development, because we have the expertise and we can generate the necessary financial pile that is required to do that.
Great, thanks for that. I only see one more question in the chat, so we might be closing up with this one, and it might be a fun one for you. I don't know. What about the killer hornets?
Oh God, yeah, I don't know. It's like, I don't know what happened, because the killer hornets are around for a while. I think they have been spotted in Canada last year already. The killer hornets are present in Europe and have spread through Europe. So it's not that it's something that completely came out of the blue. I think several Asian countries had problems with the Asian hornets. Obviously, some journalists hit the right kind of title, the killer hornets. Right? They are not killer hornets, right? It's like killer bees, right? It's like, I don't like the naming of that animal as a killer hornet. They can be a nuisance. So we know them from Europe. I've seen them in Asia, because they go for the colonies, right? So the Asian bees are tolerant against the Asian hornet because they form a ball around the hornet and cook it to death. You might have seen that. If not, there are some really cool YouTube videos on that, like how the hornets are cooked to death. The commercial beekeepers in China, they have like a queen excluder in front of the hive, but the queen excluder is a bit bigger. So the bees can go in and out, including the queen, but the hornet is too big. So the queen, the hornet doesn't get into the hive. And from what I understand, that actually, by and large, actually fixes the problem. And the hornets, they're not kind of chasing humans, right? If you have a hornet hive in your backyard and you think you can climb up with a spray from Lois up there and kill it, then you're probably in trouble, right? You shouldn't do that, right? But it's like these animals are not out there to kill us. And I think there were kind of attempts from our end to kind of put a little bit more reason into that. I think the killer hornet, and that's what they probably do, they should monitor what's happening up there, whether they get established or not. But from what I understand, I think there are 2 observations, a dead hornet and I think 1 live one. But whether they really get established up there, that is probably something that is, at least from what I know at the moment, it's like, it's unclear.
Boris, it's Washington State University, their apiary department. They have asked all of the beekeepers and everything up in the Washington area, because they were found way north up by the Canadian border.
Yeah.
And they did find 2 of them. And so they are currently setting out traps and asking other beekeepers and anybody else around to help, you know, because they do want to find out if they're colonizing or what's going on, or if it was just a fluke where they came in on a ship, you know, from somewhere.
Yes.
Up there at the Canadian side or Seattle side. So, but yeah, I think it was the media, you know, but yeah, they did find 2, but that's up in Washington at this point in time. So they're
and that would make sense because they have observed them. I think at some point last year they were spotted in Canada. Right?
They were in Canada.
That's why it came out of the blue, because they have already been there. Look, it's like it's an introduced species, and that's not what you necessarily want, right? Nowhere. So it's not. I don't say this is harmless, and it's like it's no problem. They could cause ecological havoc, and in Europe they're really unhappy with them. Well, I want to avoid like that. Like there's some like realistic kind of recognition of what that animal is and what it isn't. I think that would be fair from a scientific point of view.
Yeah, I think the biggest danger is to the feral colonies, because there's no protection against them, whereas beekeepers, you know, we can put entrance reducers, we can do all kinds of screens and whatnot on our colonies, but it's the feral colonies that they're afraid of, that, you know, something will happen. But
there is no question that these hornets could be very bad for native bees or bumblebees if they find them.
Yeah, and they are using those glue traps and all kinds of stuff. So, yeah. But haven't seen anything on it lately since then. So it's kind of just hit the news, and now it's kind of gone wayside a little bit.
That's interesting that you bring up the hornets, because when we keep bees here in Riverside, towards autumn, our hives are filled up with wasps. And we actually started 1 project now, because we're worried, like they're really crawling around on the combs, and there's not one, there are like dozens of them per frame of these wasps. And we have 2 worries about that. So they nick the larvae in autumn, so I don't really like that too much. And the other thing is whether they drift between hives and whether they can transfer diseases between hives. So we have like 1 project lined up to look into that. I don't know whether that is just something that we see here in Riverside, or whether you have seen that as well.
Yeah.
If they transmit diseases between hives, then that's I don't like that at all.
Well, hopefully we can just keep them out of the hives.
Yeah, I agree. And like we do research on our hives, but we don't have commercial hives, so we're not stressing them out. We don't put them on pollination contracts. So generally, my impression is that our hives are actually pretty happy, right? So we have about 80 hives, and we rotate them. So 40 are used for experiments, and then we have the Club Med area, and that's where we place them, and we feed them, and they get everything they want, right? Just so they are as happy as Larry. And so like our bees in general should be really happy, but we see quite high infestation rates of these wasps, and they steal things. So they direct the larvae out. I don't know whether they go after the honey, and there seems to be some difference that some hives seem to attract more wasps than other hives. But I don't understand why or whether there is like a problem with the guard bees that they don't recognize them. But yeah, that's a project. We haven't found a student yet, but I might come back to you if you get a student on that we could, for example, sample wasps from different hives, and we have these genetic tools where we can screen what type of diseases they carry in them. From other parts of the world, we know that that the wasps, that you find bee diseases in these wasps. What I don't know is whether they transmit them, but we can see that the wasps carry the diseases, which is maybe not too surprising if they eat larvae and if they eat bees. Huh?
Yeah. Open for the future.
Yeah.
All right, Liz, one more question. And then I'll let you close it out.
Yeah, I think that's pretty much it. You know.
Sounds good. Sean, let's go ahead and have you won the last question of the night. There it goes. We're unmuting each other. Go ahead.
Okay. For those who want to keep commercial queens, do you have any hypotheses as to why the commercial queens keep being replaced so quickly in your hives? Do you think they're poorly mated, that they're just doing a supersedure, or do you think they're getting invaded by a feral hive and having a queen snuck in and they're killing the commercial queen?
So we observed that now for 2 years. Unfortunately, we don't have hardcore data on that, but our impression is that there's a couple of factors that play into that. The first one might be that, like, if you bring in queens from other regions, that they are, like, Riverside is pretty tough climate-wise. It's like glowing hot during the summers. It's like desert-like climates, right? So I don't know, like, how happy a Hawaiian queen or a queen from the north really is in that environment. And there are now really good data, and there was an entire symposium on that at the last Apimondia, that the queens are relatively heat tolerant, relatively, but the semen inside the queens in that little pouch is extremely fragile. And we found that in Australia as well. So, like, you increase the temperature of the queen only by very few degrees, and the queen's running around is happy, but the sperm's all dead. And that we feel that could be an issue. So because we deploy these sensors now into our hives, we see that, like, the temperatures in the during these hot days where you have over 100, 110 degrees Fahrenheit, right? The temperature inside the hive is just really, really high. And what they basically said at the Apimondia is that over time, the beekeepers, to save costs and to have more space, they basically reduce the thickness of your gear. So that also means you have less insulation inside the hive compared to outside. So they, for example, at Apimondia, they recommended to use boxes, if you have wooden boxes that are thicker, just to kind of provide better insulation. So that I think, like, that could be a real issue because we lose them so fast. It's a bit scary. And I think one thing that we also, and I think we have pretty good data for that, is that if you have a requeening event, the more Africanized or the more kind of non-managed genetics you have in your hive, the less eager they seem to accept the queen. That's just our experience. I don't know whether you have seen the same.
I run all feral hives, so yeah. I don't keep track of whether or not they're commercial or not. But I'm thinking for the people within the clubs that are in a residential area, they obviously want to avoid having any feral bees in there to keep them really nice. One of the things that I'm thinking of, Eric Musson, who was the previous state apiarist, did a research project where he tested the viability of the sperm before and after they transported them. They put sensors in with the bees as they were transporting them, and they found that they were subjected to both extreme hot and cold during transport, you know, going up in a plane and then getting down on the ground in some hot truck. And as a result, they wiped out a lot of the semen as a result of that. And that's part of the reason they're saying that a lot of the queens will have to be replaced, you know, oftentimes once or twice a year because of that.
I have a beekeeper now, and I think we requeen every 3 weeks, right? And it's like the success rate is nowhere where we want it to be. We just need these managed queens because we have like, for the bees and beekeeping class, we have students, all of them are non-scientists, non-biologists, so they are dancers and architects and engineers, right? So we need manageable bees for them. Otherwise, like, there will be not a long and happy relationship between the bees and these. So we buy like almost, I don't know, once a month or twice a month, we buy these queens and bring them in. It's really frustrating. So my beekeeper, which is it's Jamie Scholar. I hope you all meet him at some point. He basically also came up and said, like, we need like local queens, because that's not really going to work. And we see the effect, right? Even if you still have maternal genes, the first kind of takeover, so where the mother queen is replaced by a daughter, we almost instantly see like these bees doing much better. So again, yeah.
If you take and artificially inseminate your own commercial stock, do you get a queen that will last longer? And do you find that you lose more of them when it's really super hot out?
Well, unfortunately, we lose them all the time. So we collect all that data. We have done artificial inseminations last year. Our AI queens lasted longer, or okay. To be honest, we used the last year to just set the entire system up here. It's not, you have to kind of, you need like a very sophisticated timetable from when you graft the eggs, that the males come in. You have to, the tricky thing with artificial insemination is to basically prep the hive so that it accepts, that the hive accepts a new queen, right? So it's, and that seems to work a little bit differently here in California than it does in Australia. So we don't have like like years of experience of that, but we got it to work. And I think, like, from what we have seen, that seems, at least in the beginning, the way to go, right? And that's why we basically came up like on our end by saying, like, we need like a queen breeding program here, because this is not going anywhere. And I think wherever I show up and raise that idea, I think, like, I get a lot of people basically nodding with their heads and say, like, we need something local here that basically that we can keep here. And so I think, like, that's actually quite a nice activity to go forward. So we will AI again. We have set up another round. I don't know when. Jamie's working on that. I think that will happen. Teaching is coming to an end next week. And then, like, we have the summer break, and then during the summer break, we just run a second round of experiments. And I don't think at the moment we are really after like a queen breeding program. We basically just want to have like all the setup and all the methodology in place, so that we know, okay, if we do it like that. In Australia, we had a success rate of 90%, right? So, and that's what the Germans are, I think, at 96%. And from what I know, for example, the Polish beekeepers, their entire stock that they maintain is all artificially inseminated. So you can produce a lot of queens, and you can basically run stock through that technology, but you have to locally adapt it to the bees, and also to the climates and to the seasons.
Okay, thank you.
Sounds good. Trish, did you want that question answered? It's after 9 o'clock now. So okay, the last the last question, any research on varroa treatment effect that it has on drone sperm?
Any varroa?
Any research on varroa treatment and effect it has on drone sperm?
We haven't done anything. I think other people have worked on that. I actually would have to look up what's coming out of that. I know that like there was a study on that, and I think it showed that like that the males were unhappy with. Karine, do you know that? Or anyone else? You can't talk. You're all muted. You all have to listen to me. That's the power. I think there was.
Oops, sorry.
Yeah.
Just like you, Boris, I read this article. I read this scientific paper. I don't remember the year, but yes, it does affect the sperm of the male. Yes, it does.
Yeah.
We can find it again, I guess, on the internet, probably.
Yeah. What we found in our research is that the males are like they are little wussies. It's like it's unbelievable how susceptible they are.
Yes.
Like a little bit of a higher temperature, a little bit of this or that, and poof, off they go. Right? And
Just
I think
like husbands.
they are going to mute you again. You know.
That's right. I'm gonna mute myself.
I think the advantage of that is that you can use the males a bit like canaries. So if you have like males in the hive and they're healthy, that means that the hive is probably doing well as well. The only problem that we saw also in Australia, and that was some work that we did for the beekeepers there, is that the males might run around, but then when you look at the semen or when you run an AI, they're just all crap. It's like they either have dead sperm or no sperm at all, or the sperm is completely glued together, so you can't actually extract it anymore. And whenever we use that semen, you're not having any success. So that's something that like Barbara, for example, is really interested in that type on that side, right? Because if we set up a queen breeding program, breeding the males, having them at the right age, and making sure that like you have sufficient number of males available, that is an absolute key. The rest is relatively simple. So I think if we can artificially inseminate up to 100 queens a day, right? So if that machinery is running, you can be very productive, but you need the material, you need the animals at the right age, and they need to be tip-top shape. Otherwise, either you don't get the semen out, or the males become the queens become drone layers, or they're never accepted back into the hives.
Okay. Okay, well, we're going to close it out for tonight. Boris, thank you so much for all the great information. We had a really good turnout tonight. We still have, 24 people, which is really good. Our meetings usually do run long, We like to give our guest speaker as long as possible, because it's good information for everybody. But again, thank you so much, everybody. Give a hands up, you learn something new, but Rick's computer froze. Thank you, Rick. But yes, thank you so much. We will definitely be in touch. We were already talking amongst a few of us of trying to find some place to where we can get everybody together to watch that film and get it on a big screen. So we're going to be looking into that hopefully as soon as everybody opens up. But yeah.
One of the requests from the other beekeeper clubs was whether we can organize a viewing here at UCR. And so that's on the list as well. It's like, I just don't know when it happens, right? But if there would be interest from your end as well, that we show you the different bee yards and like the setups that we have, we could organize something like that as well. As I said, it's out of my hands. So probably not this year. That's what my prediction is. But maybe it's
still push for this year.
Sorry?
I said we can still push for this year.
Well, let's hope. Fingers crossed, right?
Yeah.
Yeah.
Well, it depends on when they let groups meet again. So that's the big biggest factor. So,
And maybe at some point we also get some positive news again, because that would be really helpful to have something positive again.
definitely be in touch soon. So as soon as you find out anything about any of the grants, if you need any other letters or, you know, you have to support the Orange County Beekeepers Association. So I want to let you know that. And everybody has your web address, and they can go and go on your Facebook and everything else on. So keep in touch.
Good.
So thank you so much.
What a pleasure. Okay.
Okay, everybody, we're going to close it out. Thank you very much. The next hangout is June 17th, and you'll get notified of that as well. So same time, 6 to 7 p.m., same Zoom information, but you'll get it sent again. So good night, everyone. Be safe, and we'll see you later. Thank you. Good night.